Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12 Molecular Basis of Inheritance, that is Part 6, that is RNA World. RNA World. From foregoing discussion, any immediate question becomes the evident which is the first genetic material. So, what is the first genetic material? It shall be discussed in the detail in the chapter the chemical evolution, but briefly we shall there hide the sum of the DNA faster in some few points. So what is the first genetic material? First we discuss about it in a few points here only. First, the RNA was the first genetic material. RNA, the ribonucleic acid, that is a first genetic material. There is no enough evidence to suggest that the essential life process such as the metabolism, translation and splicing, etc. So they have no evidence for as in that the process of the what metabolism and translation and then splicing process, etc. also. And mostly in this they are complete this total process evolved from where in that RNA. So basically the total the genetic information we are obtaining from where in that only for RNA. Here RNA use their act as in the genetic material as well as in a catalyst. In some they are important the biochemical reactions, the living system that act as a catalyst by RNA catalyst and is not a protein enzymes. So mostly they get act as in what? Get act as in their catalyst. Next, here, but RNA here beginnings here the RNA begins the catalyst was the reactive and hence is in a unstable. So it is completely their unstable genetic material. There, therefore, the DNA has evolved from RNA with as in chemical modifications that make as more stable. So the mostly the completely the complete the DNA evolved from where in that RNA only. The DNA being the double standard having as in a complete the complementary the stand for the resistant change evolving a process of any repair. So for example, when we take as in this total the DNA, within the DNA the cell repair will be there takes place. Let us here the replication process. Here what is that in a replication? Replication. See, while the proposing that in a double helical structure of for the DNA, the Watson-Crick and had immediately the proposed the scheme for as a replication of the DNA. So the scientist was whom here their first the Watson-Crick. Here the scientist was in a Watson and their Crick. Their first they are discussed about as in this DNA and immediately they explain about as in the scheme of the replication. It means immediately when they explain the total DNA, immediately there he completely explained. Now the quite there in the original the statement for they get follows. So here what is that in exactly the replication? Let us be discussed in a one by one process. Okay, let us next see it has no the escaped for our the notice the specific the pairing we have in the they are postulated immediately suggested the possible copying of the mechanism for as a genetic material that Watson and Crick proposed as in 1953. So mostly the pairing are completely they are postulated immediately suggested they are possible copying of genetic information. Means here for example it is in a RNA strand. So in this RNA strand they get completely get making as what incompletely one DNA strand. Am I right? And it is the proposed scientist was whom? Their Watson and Crick. Okay, let us the scheme of suggested as any the two strands would be there separated, they act as a template to synthesis of as the new complementary strands. So for example, when we take here, it is in a that these strands are called as these both strands are called as what? These both strands are called as in template strands. These are called as what? Template strands. They are completely they get participate in the DNA replication. They get participate in where? In the DNA replication process. And here after the completion of the replication, each DNA molecule would be having a one parental and one synthesized strand. So when they get forms here, what happened? It is in a one strand, it is in a one strand. Am I right? One is completely the newly synthesized form. One is in a newly synthesized form. One from as in a completely that is in a parental strand. Now here this the scheme was their first their semi-conservative the DNA replication. So it is their first their explained this is called as what in a semi-conservative replication process. Okay, next here what is the experimental proof? What is the experimental proof? The experimental proof, now it show as in the proven the DNA that replicates the semi-conservatively. The DNA to get replicates as in a they are semi-conservatively. It was the first they're showing that Escherichia coli. The first they are noticed in which organism? That is in a Escherichia coli, and subsequently their higher organisms after they get they're noticed as in higher organisms like as in plants and animals like that organisms. Okay, and here the first higher organisms like as in plants and human cells also. The scientist was the Matthew Smelson and Franklin Stahl. They performed as in their following experiment in that in a 1958. So scientist was in the Matthew Smelson and as well as in the Franklin Stahl. They proposed as in they give as in that experimental proof. What is the exact that? What is exactly the genetic material DNA? Okay, now first, the scientist was they took as a name one culture plate. He took as a name the culture plate. He was grow in that Escherichia coli on the medium. So these are completely called as in what E. coli cells. See, they are growing the E. coli medium contain. They are completely NH4Cl N15. See here that N15 contain medium that is the NH4Cl ammonium chloride that is in a N15 isotopes. There N15 is in a heavy isotopes the nitrogen. So nitrogen contain as in what? In a heavy isotopes. As then only the nitrogen source of the in the many generations. So here what happened in this completely in this N15 they comes as in that many generations. Am I right? These are completely they comes as in many generations. That result was that in the N15 was incorporated into as in the newly synthesized DNA as well as other nitrogen contained compounds. Here they are completely N15 they get participate in the synthesis as an DNA. Am I right? After it is the nitrogen based compounds also they get participate in this in a medium. Now this heavy DNA molecule could be there distinguished from as an that normal DNA the centrifugation with the help of in cesium chloride. So now in this medium they get change into, for example, this is an N15 medium, N15 NH4 the Cl N15 medium they get transferred into as an that NH4Cl that is an N14 medium. Yes, now here they require as one mediator. Here they require as in what? Here to require as in one mediator is required that is called as what? That is in a cesium chloride. Every 20 minutes here the every 20 minutes here they complete the as in the cesium chloride. Every 20 minutes they get the medium will be get changed. Am I right? Let us after the density the gradient the please note that in a N15 is does not the radioactive isotope and it can be there separated from as an N14 only the based on the densities. So here N4 then N15 is there and then N14. The N15 is not completely they have any heavy isotopes because they are completely the density will be there. They're changing here. Okay, let us after when they here then they transferred the cells into the medium with a normal NH4Cl and took the sample with various the definite time the intervals the cells they multiplied the extracted the DNA the remained as in the double stranded helix the various samples where they separated the independently with help of the cesium chloride gradient the measured with the densities in the DNA. So for example here they have as an the N15 isotopes are there, right? Here in this N15 isotopes when they get go to as in the generation to generation every 20 minutes here generation to generation what happened they get complete they get participate in the replication process. Okay, here the mostly the NH4 there the NH4Cl is took samples in various the time interval periods means here every 20 minutes we are complete they were taking as what samples. Am I right? That is now you can recall what with the centrifugal force is in that think why as in a molecules with as in a higher mass density would be there completely the sediment faster because here what happened we are using as that mediator is what that is in a cesium chloride. Am I right? So then already these heavy isotopes are there when you are completely they are changing into as an N14 medium what happened that complete the density will be they get they are faster. Okay, that is next. So they given as a clear picture in the attaining process. It is in a yes. It is in a N15 DNA molecule. After that any gravitation force, what happened? It is they get transferred into as an N15 to N14 medium within the 20 minutes. What happened? It is complete to get formed as an hybrids. Means one is a N15 is there, or N14 is there. So they are completely incorporated. Am I right? So the two strands will be they are getting here. That is called as one is a N14 and the one is in a N15 medium. So that is called as what complete in a hybrid medium. And then after within the 20 minutes, here 20 plus 20, here 40 minutes. Am I right? After in that second generation is the first generation and it is in the second generation. In the second generation, what happened? In this completely here the N15 generation will be there completely there. The number will be there reduced because what happens? Generations will be increased. What happened? The parental genes will be they get reduced. Am I right? So then what happened here? The mostly the N15 there takes place only the 2 copies we are getting. The remaining all we are getting as an N14 means here what happened? Here mostly here the light DNAs means light DNAs are nothing but what N14 the DNA particles will be there increased for this medium and N14 will be increased and N15 that is also what in that part of the N15 N14 that is what that is a part of the hybrid medium, right? In this hybrid bit they comes as an A, that's in second generation. In that we go to a third generation, what happened? The third generation they completely the N15 is complete they get absent, only we getting as an A, N14 their DNA fragments we are getting. Okay, now this is completely called as what? The separation of the DNA by process of a centrifugation process. They explained by the Michelson and Stahl experiment. And then after see this DNA was there extracted from as in that culture one generation after then it transferred into as an N15 to as an N14 medium that it was in every 20 minutes. So this total process will be takes place in what? In every 20 minutes we are doing. The Escherichia coli divide every 20 minutes and as in a hybrid or intermediate the density. That every 20 minutes they are completely what happened? The first having heavy DNA is converted as a hybrid DNA is converted as a light DNAs. Okay. Now here the DNA extracted from as in the culture after another generations that is in after 40 minutes means it's in a second generation was mostly they are composed of an equal amount of the these hybrids DNAs will be their light DNAs are comes. Okay, that is next. If E. coli was allowed as in a grow in 80 minutes, what would be the in a proportion of the light DNA hybrid densities in the molecule? Already just before only I said what here automatically the generation will be increased. What happens? The more light DNAs we are getting. Simply, yes, na? The more the light DNAs are comes. Next, the very similar experiment, the moving using as in the radioactive thioamine to the detect as a distribution of the newly synthesized DNA, the chromosomes was in the performed as in a VCA faba, that is in a faba beans. So they completely how to their semi-conservative process will be going on in the same process here. There another experiment or the data that some scientists that is in a Taylor and his colleagues in 1958 in that species is what that is in a faba beans, that is in a VCA faba the faba beans. It did as an experiment. This experiment they proved as in a DNA in the chromosomes also in the replicates in semi-conservatively. So mostly the DNA fragments here, the DNA fragments also to get participate in that in a process of what? In a semi-conservative process. Here the semi-conservative processly they get participate and to get produced as what? In a new stance. Okay, this is in a completely that semi-conservative process, the genetic material. Okay, the remaining to discuss in next class. Thank you for watching. Please share, subscribe and like.